Thank you very much. I'm delighted to be here. It's a homecoming of sorts because I was in graduate school and medical school at UCSD. And speaking of that, I'd like to throw at you at the beginning a sort of geriatric medical mystery. Hudfreeze and I started in graduate school the same year, more than 45 years ago. And I evolved into the gray beard that you see before me, but Hud still looks like a young Robert Redford. So if anyone has insights into that mystery, please see me at the break. I was at the FDA for 15 years. I was sort of their biotechnology czar at the beginning of the introduction of bioengineered drugs in the 1980s. I've been away from FDA for 20 years now, but I continue to study it from my perch at Stanford. Regulators are not comfortable with many new technologies. And however excellent the science may be, and however avid the advocacy, and we've seen great examples of both during the past day and a half, regulators will inevitably become involved in the end. And you heard that in some of that in the previous talk. Part of what you need to know is to understand the psychology of regulators. And that gets to the dichotomy between what the risk analysis community calls type 1 and type 2 errors. A type 1 error is a regulator approving something, permitting it to be used that proves to be dangerous. So the prototypes of that are thalidomide and the swine flu vaccine in the 1970s. And when that happens, the sky descends on the regulators. There are congressional hearings. They're excoriated in the editorial pages of the New York Times. And so they quiver at the thought of making a type 1 error. A type 2 error, on the other hand, is not permitting something important or useful to be available. And that's manifest by delays, by not proving a product at all, by asking for yet another clinical trial or another 50 monkeys. But a type 2 error is a non-event. Almost no one knows about it except the company that's involved in the trials and maybe a few Wall Street analysts. But there are no consequences, essentially, for regulators. So they'll make type 2 errors in order to avoid type 1 errors. And we've heard a couple of people allude to the phenomenon of rare diseases that affect only a few hundred or even a few thousand not being taken seriously by funders and by others. But regulators often show what I would consider a callous disregard, even for very common, very serious clinical situations. Let me tell you about the genetically engineered rice from a company called Ventria BioSciences. They wanted to come up with a way to treat diarrheal diseases, which is the second leading killer of kids under age 5 in developing countries. And so the common treatment for severe diarrhea is oral rehydration solution, which is essentially a mixture of water and salts and sugar. And sometimes it works, sometimes it doesn't. That depends on the baseline health of the child and the severity of the disease. But what these scientists found out is that rehydration solution is markedly enhanced by the addition of 2 human proteins, lactoferrin and lysozyme. These are natural proteins that are found in human breast milk and in tears. And Ventria genetically engineered rice to produce these, to use them as mini factories, if you will. So the proteins are purified from the rice kernels and added to rehydration solution and have a marked effect. reduces the length of time of the symptoms of the diarrhea and also reduces the likelihood of recurrence. What this required from FDA was simply an affirmation that it was generally recognized as safe, GRAS. And this is done traditionally on the company convenes a panel of experts. They make a recommendation which is transmitted to FDA, and FDA grants the affirmation. FDA never responded to this. So after almost a decade, Ventria is not willing to sell it without FDA's affirmation, and so it's not available. Golden rice is another example, although it's not an FDA example. It pertains to regulators abroad. Golden rice is enhanced with beta carotene, the precursor of vitamin A. Vitamin A deficiency is a scourge in much of the world. It's critical for normal vision and helps maintain the immune system. 250 million preschool children are vitamin A deficient around the world, primarily in countries where rice is the primary source of calories. But rice doesn't contain vitamin A, and often the diets of these kids don't provide vitamin A in any other way. Of those 250 million, 500,000 go blind each year, and within a year of those, half die. The blindness is kind of a marker for general severe debilitation. Golden rice contains therapeutic amounts of beta carotene, and it's been shown in clinical feeding trials that it does prevent deficiency. And this product has been in regulatory limbo for more than 12 years in places where approval has been sought: the Philippines, Thailand, and elsewhere. Regulators, some of the regulators at places like the UN, the Food and Agriculture Organization, and others have not been helpful in the least. Now, I want to move on to the real subject, but what I'm trying to tell you is that regulators often say soothing things, but you have to keep real pressure on them in order to realize actions. I want to talk about the promise of gene therapy, which can be of two types: somatic cell or germline. And so somatic cell gene therapy, some of which you've heard about, involves manipulation of gene expression in cells that will treat the patient but not be inherited by subsequent generations. And so, for example, in the last talk, stem cells are an obvious choice. Somatic cell therapy modifies only non-reproductive cells, and it's generally been uncontroversial except for discussions of enhancement, modifying normal kids in order to make them jump higher, run faster, be smarter. And this is kind of a model for how that's done. Up here you see a new gene introduced into viral DNA, in this case adenovirus. The virus then attaches to target cells in the body or perhaps in lymphocytes ex vivo, which will then be infused. And then the virus enters the cell via vesicles, and its genome, including the new added gene, is expressed inside the cell. There have been a number of trials of somatic cell gene therapy over the last 25 years for all sorts of things, and these are shown here. Most have been for cancer, about 10% for monogenic diseases, and lesser frequency for cardiovascular and infectious diseases, and very few for anything else. There have been some marked clinical successes for somatic cell gene therapy, particularly severe combined immunodeficiency, especially due to ADA, adenosine deaminase deficiency, beta thalassemia, a single gene, a single amino acid substitution in hemoglobin, Wiskott-Aldrich syndrome, a deficiency in something called the WAS gene, which is important in maintaining the cytoskeleton, and acute lymphoblastic leukemia. And this last one is of particular interest because here's the child who underwent treatment with this, and this really represented pushing the envelope. This was done at the Hospital for Sick Children in London last fall. This child was resistant to all of the standard therapies, was about to succumb to her disease, and the docs at this hospital got permission to infuse her with donated T cells that had been modified to target her lymphoblastic cells. There were genes disabled that reduced the likelihood of graft versus host disease, and also made those T cells resistant to the chemotherapeutic agent that she was about to receive, and she's still doing well. And this pushing the envelope is very important. It's made possible only by the sorts of pressures that the people in this room today exert and should continue to exert. FDA has sometimes, again, not wanting ever to be accused of allowing a dangerous drug or a dangerous trial, has twice shut down gene therapy clinical research entirely, once in 1999 and again in 2002. The 1999 incident was a particularly odd one. It was clearly anomalous. An 18-year-old boy or man at the hospital of the University of Pennsylvania was in a clinical trial. He was the first patient. He suffered from ornithyl transcarbamylase deficiency, and he was perfectly well compensated on diet, but he was the first patient. And the night before the procedure, he left the hospital and ate things that were not on his diet. The morning of the procedure, his lab values were not within the bounds of what was appropriate for the trial, and yet they went ahead with the trial. He had a massive sort of allergic reaction to the gene therapy and died, and FDA shut down all of gene therapy as a result of that. Again, in 2002, there were a couple of kids in gene therapy trials in France who developed leukemia as a result of the gene therapy, because at the time retroviruses were being used and were introduced in a way that activated an oncogene. And so FDA stopped all gene therapy, including that which used other viruses than adenovirus. So again, they're very sensitive to negative results. Now, germline gene therapy, distinct from somatic cell gene therapy, modifies reproductive cells or embryos, results in permanent changes that can be passed down to subsequent generations, and is the only feasible approach for certain situations, such as two persons with sickle cell anemia who create an embryo. Sickle cell is a homozygous recessive disease, and so all of the offspring of two sicklers will be affected. Germline gene therapy has become kind of the boogeyman of bioethicists and of docs who don't see patients. For example, Harvard stem cell researcher George Daly said last year, quote, "This is an unsafe procedure and should not be practiced at this time and perhaps never." Well, I think Dr. Daly should have a look at the kid who was treated in London and at the kids in Lynn Wolf's slide and at some of the kids in this room, and then see whether he can repeat that statement. Dr. Daly also seems not to realize that technologies are rudimentary at the beginning, but improve with use and learning and refinements. And so when I was in medical school, bone marrow transplantation was dismal. It had a survival rate in single digits. It was performed only at a couple of institutions. And then cyclosporin as an immunosuppressant was discovered and other refinements were made. And now bone marrow transplantation is routine throughout the country. Same can be said about open heart surgery and also renal dialysis. There is a learning curve and we get better at these, often much better. Now, you've heard about and you know a lot about CRISPR Cas9. This is the headline of a blurb from Nature on December 1st. More precise molecular scissors, more precise use of CRISPR Cas9. Since December 1st, there have been 3 papers that have described marked increases in the precision and accuracy of CRISPR Cas9. And so in a remarkably short time, we've gotten closer to being ready to do gene editing of embryos and of perhaps of sperm or spermatogonial cells in culture if we weren't already at that point. In December of last year, there was a meeting in Washington, D.C. of the National Academies of Medicine and Science of the U.S., U.K., and China, which effectively called for a moratorium on inheritable genome changes in clinical research. Their summary statement said that it's irresponsible to proceed until risks are better understood and there's a broad social consensus about such clinical research. The participants in this meeting were mainly people who don't treat patients. And many of us think that this was a kind of groupthink exercise. One of the participants referred to the organizers, who are extremely eminent in the scientific community, as charismatic megafauna. And her comment was that we shouldn't allow the charismatic megafauna to dictate policy to the others. So the outcome of this, I think, was politically correct. It was preordained, and there was generally a dismissal of dissenting views. Not everyone feels that way. Harvard psychology professor Steve Pinker, with whom I generally agree, said what we need from bioethics is get out of the way. Bioethics, he said, should not fetishize sweeping rubrics such as dignity, equity, social justice, sacredness, privacy, and consent at the expense of the health and lives of actual people. And bioethics should not thwart important research with sensational speculations about eugenics. And I can here for a moment circle back to that 18-year-old who died in the University of Pennsylvania trial in 1999. The reason that he was selected, even though he was perfectly compensated on his diet, was that the bioethicists at Penn, mainly Arthur Kaplan, said that it was inappropriate to begin the trials of gene therapy on kids who were badly affected and who were likely to die, because such a situation is coercive to the parents. In other words, he said they couldn't give informed consent because there was coercion on them, which I think is a simply insupportable view. We've heard a lot about the wonderful things that NIH does. And as I mentioned yesterday, I was a postdoc there and loved it. But NIH still has the recombinant DNA guidelines, safety guidelines, left over from the 1970s. And its Appendix M says the RAC, the Recombinant DNA Advisory Committee, will not at present entertain proposals for germline alterations, but will consider proposals involving somatic cell gene transfer. And they define germline alteration as involving a specific attempt to introduce genetic changes into the germ or reproductive cells of an individual with the aim of changing the set of genes passed on to the individual's offspring. Now, given that NIH, either at the level of the RAC or the level of the NIH director, can reject a clinical trial of gene therapy, of germline gene therapy for any reason, it seems to me bizarre that the RAC will not even entertain a proposal for any sort of germline gene therapy. The guidelines apply to research conducted at or sponsored by any institution that receives support for recombinant or synthetic nucleic acid molecule research from NIH. So that's essentially every academic institution that might want to do this. And therefore, a de facto moratorium is already in place. My view is that NIH should repeal Appendix M. NIH, in fact, should get out of the business entirely of reviewing either somatic cell or germline gene therapy. FDA and institutional review boards inevitably have responsibility and experience in these areas anyway, whereas NIH doesn't. And when you get NIH involved, you have not only the RAC, but institutional biosafety committees at research institutions that have to also be involved and approve this. So in conclusion, human lives are more important than political correctness or abstract notions about ethics. If you don't take the first step, you never reach the goal. And the government should be removing obstacles to germline gene therapy and certainly not creating new ones. Thank you. Glad to take some questions.
Yeah, I think, again, for the sake of time, we've got time for one.
I'd just like to say that I thank you for publicly holding Arthur Kaplan responsible for what happened to Jesse Gelsinger, for one thing. Secondly, I think that we have always asked the Food and Drug Administration to look at risk-benefit ratios instead of approaching things with risk aversion. And so I agree with all the things that you said. And then this sort of what some of the other people say about this avoidance of new things because they don't take care of patients. But more than that, they don't have children with disorders like this. And they don't have relatives with disorders like this. And if you look around the communities that have really rare disease relatives and children especially, what they sometimes hope for is that a member of Congress will actually have someone affected, which is a terrible, terrible thing to have to resort to here. But if a member of Congress does have something, a child with this, they'll call the FDA. The FDA will respond. And they will establish legislation to right some of the wrongs that you just presented to us today.
Well, since you mentioned Congress, one of the ways to exert pressure is to go and see your congressmen and senators, preferably in your district, and bring these kids along and tell them what it's like and show them what it's like.
Yeah, this has definitely been going on for the Duchenne's and ceroid lipofuscinosis community. So